right. Well, thanks for everyone joining us here to the latest edition of our seminar series. Today we have James Junker, who's currently working as a postdoc at Michigan Tech University, and he's going to be talking about a bunch of really cool work that he's been doing up in Iceland. I'm really excited to see this talk. Just a bit of background. So James has really nice CV I'm looking at here. So he did his it looks like his undergrad at Northland College in Wisconsin, master's at Montana State University with Wyatt Cross, and he also stuck around and did his PhD there. So he's spent a lot of time. I was up in Montana last week. It was really beautiful, or the week before last actually. So nice country. He's currently working as a postdoc researcher, like I said, and he's also have a visiting scientist at Louisiana University's Marine Consortium. He's super prolific, so it looks like he has about 11 published papers in really, awesome high quality journals. He's an up and coming rock star, and I just saw that he got really nice, grants and stuff like that. He's going to impress us today, talking about some research that he did. I guess mostly doing your PhD, Jim.
Yes.
Yeah. Awesome, cool. Thanks. We're all excited. Take it away.
Excellent. Thank you for that introduction, Clay, and thank you to Jess Corman for inviting me to give the talk, and thank you to the Paul and the Ratios Matter crew for putting on this seminar series and keeping us up to date on kind of the up and coming and latent breaking in the stoichiometric world. Today, I'm going to talk a little bit about thinking about kind of stoichiometry of kind of diverse organic matter pools and the environmental and ecological drivers of them, specifically highlighting how the framework of ecological stoichiometry might be able to address some of the pressing questions in ecology, specifically how we think about the relationship between biodiversity and ecosystem functioning. And this has kind of been a thrust of a great body of work over the last few decades, trying to understand how biodiversity influences the functioning and processes of ecosystems. And we can think, biodiversity is multidimensional. We can think of this in multiple ways. Probably what has seen the most, focus has been species richness effects on ecosystem function. But we also know that community composition, the species identities can play a role, as well as the relative abundance of species within an ecosystem that can have really important implications for important ecosystem processes like primary, secondary production, the amount of biomass that an ecosystem can support, kind of the cycling and coupling of nutrients within ecological processes. And while much of the focus over the last few decades has been kind of this one direction of what are the biodiversity effects on function, kind of historically, and the emerging kind of new viewpoint on this is kind of a melding of the old and the new, is that while biodiversity certainly does influence ecosystem functions, there's important feedbacks upon ecosystem function that might modify the levels of biodiversity that we see in an ecosystem. And also that both of these, both biodiversity and ecosystem processes, are modified by environmental controls that operate at higher levels. And understanding these connections is increasingly important given kind of massive changes on global scales to both regional and global biodiversity, as well as kind of the cycling of elements, the thermal regimes of ecosystems. And I think one of the powerful aspects of the framework of ecological stoichiometry is that it kind of connects, it gets the matter of the matter. It recognizes that elements are coupled and conserved through different processes, and that we can connect, what we and these biological structures that are made up of elements are important structurally and functionally for organisms, So we can start to connect processes that occur at kind of subcellular levels and draw a line through to the kind of global scale lithographic and climate global circulation processes that alter elemental concentrations in space and time, and also draw at all the processes that exist kind of within this nested hierarchy of ecological organization. And being able to kind of connect, at least, and having a kind of multiple currencies that we can draw between all these scales is really important, especially for these higher levels of organization. We think about these biodiversity ecosystem functioning relationships kind of emerge at the intersection of communities and ecosystems. And we know that the relationship between these two is really kind of the accumulation of a lot of processes that occur at lower levels. So processes like microevolution of populations, which Dr. Lemon spoke about in the previous seminar series here, to the traits of organisms that modify and constrain stoichiometric plasticity and homeostasis in relation to different environmental variables and different nutrient availabilities, onward up to kind of competition processes for limiting elements and how that alters the community composition and species performance across different gradients, and then upwards to even kind of the, at a food web ecological network level, where the patterns of and pools and fluxes of elements between and within ecological interactions can ultimately constrain the kind of the observed processes and the observed ecological function that we see within ecosystems. And I think when we look at these biodiversity ecosystem functioning relationships from this lens, recognizing that there are multiple roles at multiple levels at play, some interesting questions emerge, specifically, what are the relative roles of these different processes that operate at different levels? So what are the intraspecific and interspecific processes occur, and what are their relative effects on kind of aggregate and higher level stoichiometry? And how do they modify the kind of potential and realized variability of stoichiometry at higher levels of organization? Do they kind of work in concert to kind of drive stoichiometry in one direction along one, within one specific environmental variable? Or do they offset each other kind of in a portfolio effect kind of way to constrain the realized variability in stoichiometry at large kind of diverse organic matter pools? And we're certainly not ignorant to these questions. Some of the foundational observations within stoichiometry and within biogeochemistry, this relationship between marine ceston and dissolved nutrient availability within the global oceans kind of gives us some evidence that even on really broad scales, broad spatiotemporal scales, there might be kind of higher level constraints, and feedbacks that lead to relatively low range of variability that certain stoichiometries can take. And so this, kind of this classical Redfield ratio is one example of this. And when we look across different ecosystem compartments, such as forest litter and its relationship to nutrient availability across biomes, or soil microbial stoichiometry and soil nutrient availability across large global scales, we see similar constraints start to emerge, but also there is important systematic variation in some of these patterns across certain scales. And even the canonical Redfield ratio, we've started to recognize that there's important variability that exists at certain spatiotemporal scales. And so while we have evidence that there's certainly potentially these limiting and constraining effects and feedbacks on stoichiometry at higher levels, these questions still remain about what are the relative roles and of certain processes, and what is the total variability that we can observe in the stoichiometry of ecosystems. And so I'd like to today kind of discuss these questions and try to make connections between the environment, how its effect on biodiversity, and how that might alter the certain ecosystem functions, specifically kind of the total attainable biomass of ecosystems. And I'm going to do this using a specific example from this watershed in Iceland. This is the Hengil kind of natural laboratory. And this watershed is kind of unique. It has this really wide range of stream temperatures within the watershed. And so it's located right outside of Reykjavik, Iceland, on kind of the southwest portion of Iceland. And we see individual streams that range in temperature from a mean annual temperature from 5 Celsius to upwards of 55 Celsius. So we have kind of this global range of stream temperatures that exist in this really, really small geographical area. And importantly, lots of this geothermal influence is indirect, and so kind of the temperature of these streams is kind of mediated by the thickness of the bedrock, and so there's no direct interactions of the groundwater pools and kind of the geothermal magma chambers and stuff like that. So we have kind of a shared and similar solute concentration across this gradient. So there's no really confounding effects of elements and heavy metals or anything like that with temperature. And also importantly, these temperature regimes have been relatively stable for multiple decades. So we can kind of look at the longer term effects of temperature on stream ecosystem patterns and processes. And this is really, this is what brought us to Iceland, this gradient. And it's, we were there for this specific reason. However, during some of our initial observations, our initial walking around the watershed, we started to recognize that there were kind of other processes at play, obviously. And specifically, we were looking at some of these watersheds that have kind of these low-lying watersheds that have relatively low slopes. We kind of considered these to have relatively kind of low stream flow control, counter to some other streams in the system, which kind of have these big catchments that kind of funnel water to their stream channels, creating this kind of high flow control. And based on kind of some of our initial observations that kind of ran in the face of what we thought were the dominant processes, we thought this might be a unique opportunity to maybe look at the relative roles of these drivers of temperature and kind of landscape drivers of stream flow on, in this case, the kind of a maximum peak biomass of streams and as well as their stoichiometry. So we selected a suite of streams. These are high flow control. So we selected a suite of streams ranging in temperature from 5 Celsius to 27 Celsius here. And then we also selected a number of streams that kind of varied in their stream flow. And so here we have each of these box plots represents the standardized stage height as kind of a proxy for the magnitude and variability in stream flow within these streams. We tried to create this kind of these two counter gradients so that we could identify streams at high flow and low flow at both warm and cold temperatures. I think we had a decent job of doing so, considering what we had. And so in each of these streams, we go out and measure essentially the organic matter. And so here's one example of one of these cold, low-lying streams. And so this is a 5C stream that has, as we notice, a lot of what appears to be a lot of stuff. So we go out, we stick a core in the stream, and essentially take everything from the core area down to into the sediments. We then split that into compartments, functional groups, and then ultimately look at the total mass and organic matter, total organic matter mass and CNP, carbon, nitrogen, and phosphorus stoichiometry of each compartment. And we can add these all up to get at both the total stoichiometry of a system as well as the stoichiometry of each individual biomass compartment. And we're going to, again, test the relative influences of temperature and flow in this case. And when I speak about flow, specifically stream flow as a complicated variable. And so we took a number of stream flow metrics, combined them into a principal component to kind of collapse the dimensionality of it. And so when I speak about stream flow in a statistical sense, I'm specifically talking about these two principal component axes. And so let's think about, before we jump in, kind of outline everyone with where I'm gonna, where everyone and myself included, where we're gonna go. First, I'm going to describe the patterns of organic matter mass. So what are the patterns of total kind of peak biomass in these systems and their relation to environmental drivers and community composition. Next, we're going to look at the how carbon, nitrogen, and phosphorus stoichiometry varies across the different levels. So within biomass compartments and across biomass compartments within communities, and ultimately across ecosystems, and try to get a little bit of an idea of what the range of variability that exists within each of these levels. And the last thing I want to do is try to connect different community processes, so different changes in it, thinking about intraspecific stoichiometric flexibility, community composition. How do these different community processes relate to environmental variables, as well as how do they then drive the ecosystem stoichiometry that we do see at kind of this aggregate organic matter pool level? All right. So first, let's start out looking at just the patterns of organic matter mass across these systems. Each of these pies represents a single stream, and the amount of the area represents the total biomass within that stream. And we can see when we look across the gradient, we have this 2 orders of magnitude of total mass that exists in grams of carbon per meter squared. So pretty wide range, as well as different, important differences that exist in the composition and the distribution of species within each of those streams. And so when we connect these things to the environmental variables, when we look at the environmental drivers of them, we start out just looking at kind of the bivariate plots. Here we have organic matter mass, kind of total pool mass. Each of these black dots represents the mean value, and then the open dots represents the individual samples. And then we have kind of a smoothing spline just to kind of aid in terms of visualization. It's not a model or anything like that. And we look at those patterns across, in this case, mean annual temperature in degrees Celsius in this first plot, and then in relation to both of those flow principal component axes that we have. And on first blush, we don't see really clear patterns. There's neither one of these drivers is a really clear pattern with organic matter mass. And so to kind of disentangle these complex relationships that exist, we used an information theoretic approach. So using AICC model selection, we tested amongst these different competing hypotheses, thinking of flow, temperature and flow in additive and interactive models. And what this analysis showed us and gave evidence to is that even despite this really wide temperature gradient, flow is the dominant driver in these systems. And once we account for the effects of stream flow, there's no clear patterns that emerge with regards to stream temperature. And this becomes clear when we kind of account for the effects of these other variables and look at these added variable plots. And kind of the residual variation explained after we account for the other variables. And we can see, while there's clear and positive and negative relationships of total organic matter mass with our flow principal components, there's really no relationship that exists between temperature. And so flow is the dominant driver in this watershed, at least. Now, let's bring this in and think about the composition of these organic matter pools. And so what we see here is there seems to be this interrelationship between organic matter pool mass and the composition. And so just anecdotally, when we look across this, as well as when we incorporate kind of multivariate statistics, we see that flow is important for the composition of the organic matter pools. And generally, these patterns emerge that on systems with high organic matter mass, they're kind of dominated by these macrophyte and bryophyte species. In some cases, this Fontanellus in this black box here, or this black eye here, this Fontanellus antiparetica, this bryophyte, accounts for about 70 or 85% of the total biomass of that system. Contrast this with these lower organic matter pools in streams. We see that these are dominated by these epilithic and detrital organic matter pools. And these changes in the composition have important stoichiometric consequences. So now when we look at, at a broad scale, looking at the scaling of nitrogen in the closed dots here, or phosphorus in the open dots, with total organic matter in terms of grams of carbon per meter squared, we see that a few things emerge, and this is the total organic matter pool. A few things emerge. First, we notice that these exponents are below 1. So in this kind of log-log space, these exponents that are below 1 suggest that there's kind of nutrient dilution with increasing carbon mass. So nitrogen and phosphorus do not scale isometrically with carbon. And similar, or also, these exponents are pretty close to each other, suggesting that nitrogen and phosphorus mass might be relatively similar across these wide ranges in organic matter pools. Now, if we break this out into our two kind of broad categories of organic matter mass, thinking about this green kind of living biomass on the left here versus the brown kind of detrital biomass pools, we see that similarly, the living biomass and the green biomass shows this similar nutrient dilution as we get to larger and larger carbon standing stocks. Counter that, now these brown organic matter pools, these exponents are closer to 1, and they're often overlapping 1. So suggesting that in detrital organic matter, we see this isometric scaling of N and P with carbon. And so the patterns that emerge at the ecosystem level are at least shaped in part by the relative distribution of detrital or kind of living biomass within the system. All right, so thinking about now how these broad patterns emerge from kind of lower levels of organization. So looking at the patterns of CMP stoichiometry within compartments, within communities, and across ecosystems. First, let's start out with within compartments, within and across compartments, because this really represents the range of potential stoichiometry that an ecosystem can inhabit, right? So if we think about a monodominant system that exists, that is entirely made up of a Fontanellus bryophyte with a stoichiometry of 50, the ecosystem stoichiometry is going to be 50. So this sets the bounds, upper and lower bounds of stoichiometry. And so each of these violin plots represents a different ecosystem compartment. And I've kind of drawn this divider and using a little bit of this color line on the bottom to kind of separate what we consider the green components versus the more detrital components. And we see, looking at carbon to nitrogen ratios in molar in the top, carbon to phosphorus, and nitrogen to phosphorus on the bottom. What we notice is that there's both wide variability amongst the different compartments, and there's differences in the variability that exists within a compartment. And so when we think about cross compartments, carbon to nitrogen exists kind of in 5 to 90 range. And in order to kind of, I guess, standardize what this range of variability, we took the coefficient of variation of each of these stoichiometries for each of the compartments. And what that shows us is that while we have, while C to N kind of has the lowest average coefficient of variation amongst all the different stoichiometries. There's certainly differences in variability. N to P has there's variability in the variability of the N to P stoichiometries amongst compartments. But this represents single specific identifiable compartments. What do we see when we look at communities, kind of this amalgam of all these different species put together? Here we have, in a similar orientation, C to N to N to P within each of the different boxes. And now each of these violent plots represents a single stream. In this case, we're orientated from highest biomass to lowest biomass. The names of the streams aren't necessarily relevant, but what we see here is while there's lots of variability. When we again standardize that variability using the coefficient of variation, we see that for all stoichiometry, on average, these communities are more variable. When we just look at the distribution of stoichiometry amongst community members, treating all members equally in this case. And so we know that there are likely massive differences in relative abundance and relative biomass of the different species. And so now what we're going to do is kind of weight these stoichiometries of each of these components by its total mass in the system, or its relative mass in the system, to kind of get an aggregate ecosystem stoichiometry, or aggregate or organic matter pool stoichiometry, and compare the kind of range of variation to these different components. So when we do that, we see these box plots represent the aggregate stoichiometry of all the different samples that we have within each system. And we see that despite this really wide variability within community members, or among community members, that the stoichiometry that exists of the ecosystem, kind of the aggregate pool, is much reduced. And this is, again, clear when we look at the coefficient of variation of stoichiometry amongst ecosystems, that for all CN and P stoichiometries, the variability that we see at the ecosystem level is much reduced relative to species and especially communities. All right. So now that we've kind of viewed and looked at the important or how variability exists amongst the different compartments, let's try to connect this back to our initial observations that looked at flow as kind of a dominant driver of organic matter mass, and its importance for community composition, and try to connect the different changes in what we'll call biodiversity and different processes that affect the total organic matter pool stoichiometry. And so we're going to look at, in this case, what processes, what biodiversity changes drive ecosystem organic matter stoichiometry. What is the relative importance of intraspecific plasticity versus changes in community composition, et cetera. And to do this, we're going to use an application that's been recently kind of applied to biodiversity ecosystem functioning work, and that is the price equation. And so this is, the price equation was originally formulated to kind of measure and partition out the effects of maternal effects and selective effects in evolutionary contexts. And it's been kind of championed recently by Jeremy Fox, and looking at its effects on, using it to connect biodiversity loss to losses in ecosystem function. And it's a mathematical partition, and so it kind of splits out the multi-dimensional aspects of biodiversity into kind of these unconfounded and additive components. Importantly, while they may be unconfounded, they're not necessarily uncorrelated, and so there might be some interesting aspects where correlation amongst these different compartments represent kind of resistance within an ecosystem to constrain stoichiometry within certain bounds. And secondly, it operates kind of in the units of measure, and so we can take and estimate the price components and their effect on grams of carbon in the system, the total grams of nitrogen, or in this case, the C to N stoichiometry of the system. And just to break down real quickly what the price equation is, price equation is really made up of, I think, 3 distinct parts, which the third is then broken up into 3 other parts, where it looks at the pairwise differences between a system. So this delta X, so it's going to partition the changes between 2 ecosystems into these 5 components. The SRE and the SCE represent this species richness effect and the species composition effect. Really, that can be simplified to the gain or loss of species. And beyond that, every species that is shared amongst ecosystems gets encapsulated in this context-dependent effect. And so this context-dependent effect represents changes within a biomass compartment or a species that is shared amongst 2 ecosystems. We break this down into 3 different components itself. First off, the relative biomass, so the changes in the relative biomass of a certain component. The functional changes, and so this, we can think about this as the change in elemental content of a biomass pool or species. And then lastly, this interaction. So when relative biomass and elemental content change in concert, those differences get kind of shunted into this interaction term. And so we're going to use this price equation to kind of partition the patterns of biodiversity change and their effect on, in this case, the carbon to nitrogen ratios of the organic matter pools. Importantly, we're going to compare the high organic. In this case, the price equation, we have to set kind of a reference and a treatment stream. So kind of standardize, I set, we're always comparing the high organic matter stream to a low organic matter stream. That might explain some of the results in the context. And we're going to focus on the carbon to nitrogen ratios. And there's a few reasons why I chose this. We could do this with multiple stoichiometries. But the reason I think C to N is really interesting. First, nitrogen is what we believe is the limiting nutrient in these systems. I think there's strong evidence of that case, really low nitrogen to phosphorus ratios and the dissolved availability. And also, if we put the range of variability in carbon to nitrogen ratios that we see in the aggregate pools to a little bit of a context, we see that even though carbon to nitrogen, the coefficient of variation of the ecosystem pool is actually the lowest out of all the C and P stoichiometries. In the context of, for instance, we can plot it against data taken from Elser et al. 2000, where they look at Ceston stoichiometry. And here I've highlighted the range of stoichiometries that we see in the ecosystems. And we can see it's not apples to apples necessarily, because there's Ceston versus these kind of complex macrophyte, bryophyte dominated systems to epilithic systems. But we can see that there's a relatively wide range of carbon to nitrogen ratios that we see relative to that observed in Ceston globally. And when we look at and think about the environmental drivers that are important for carbon and nitrogen stoichiometry within the system, we again can use our information theoretic approach, looking at the 2 top models. In this case, we see that, importantly, flow, which has been a consistent theme throughout the talk. Flow is the dominant driver in this case of, I should say, flow and flow and nitrogen availability are the kind of shared dominant drivers of these of C to N ratios across systems. And they're really there's enough similarity and support amongst the 2 that we can't really distinguish which is the dominant driver. And so these two kind of have equal support as major control. And so we're going to eventually come back and focus on these two drivers, these two environmental variables in a second. But first, I want to kind of summarize and look at the kind of broad level effects of each of these price components on their effects on carbon and nitrogen ratios of organic matter pools. And here we have, in this case, we're just looking at the difference of, or the importance of, in terms of units of carbon to nitrogen ratio. So how each of these price components influence the carbon to nitrogen ratio of organic matter pool. And here we have this, in this case, we've combined this SRE and the SCE to kind of look at the net effect of species gain and loss. And then broken up this context-dependent effect into its individual components, the relative biomass, the elemental content, and the interaction. And what we see is that, first off, the gain and loss of species in this case has its wide-ranging effect. It has a variable effect. So community turnover is variable, but on average, it's a relatively neutral, stoichiometrically speaking, relatively neutral effect at a aggregate kind of average level. And also that when we look at each of these context-dependent influences, they all seem to drive the CETA-N stoichiometry of the ecosystem down. So they all seem to, they have stronger effects than a SRE on average, the species gain and loss on average, and they all seem to lower the CETA-N ratio of ecosystem compartments. And importantly, the magnitude that we see is certainly much greater than the magnitude that we see in terms of variability amongst ecosystems in terms of their CETA-N stoichiometry. So we see this gain and loss component has an effect that could range from -15 to +20 CETA molar units in terms of the CETA-N ratios. And so there's obviously some aspects of potentially compensatory effects going on that constrain the variability of organic matter pool stoichiometry across these systems. And so this is it's kind of a first blush at kind of trying to first go at trying to visualize some of these patterns. So I'd really love to hear if there's any comments or observations about better ways to visualize this. But I think there's some interesting patterns that emerge once we start to break down how each of these variables relate to the important drivers of CETA-N stoichiometry in the system. In this case, our change in stream flow and our availability of nitrogen. And so here we have one example and one attempt at trying to visualize and quantify these things. And so this is the effect of species gain and loss on this kind of surface, represents kind of its effect in relation to increasing or in relation to differences in nitrogen availability as you move from this first axis upward, and in relation to coefficient of variation of stream flow. And so this is one of the major drivers of stream flow that in terms of one variable that kind of encapsulates its effect on species composition and stuff like that. So this is one example to look that as we, for instance, increase the coefficient of variation in stream flow. So as we increase its flow variability, that's where we see that this gain and loss of species really has the largest influence on the kind of ecosystem stoichiometry. And that doesn't necessarily mean at these higher levels or these higher differences in stream flow that there's not necessarily species loss and gain happening, but just that it seems to be relatively stoichiometrically neutral. Similarly, at these kind of moderate nitrogen differences is where we start to see species gain and loss driving CETA-N ratios down. And it's unclear how this kind of operates. We're running up. We have a relatively small difference in DIN concentrations, and so if this relationship is really unimodal, kind of concave down like that, versus what it looks like out beyond the bounds of our data would be interesting. But to place this now into the context of all these other drivers, so when we now look at each of these different, in this case, the species gain and loss effect versus the change in relative abundances of shared organic matter pools, or the change in function of organic matter pools, or that interaction, these relationships are certainly complex. And I'm still really trying to reconcile some of these patterns with what we see at a broader scale. But we see that part of this strong decline in CDN ratios that occurs from the loss or gain of species with increasing coefficient of variation of streamflow. Part of that is offset a little bit by the increase in the relative abundance of important stoichiometrically distinct species that kind of drive this CDN ratio back up, as well as there's certainly, I think, interesting, but I still am a little bit wrapping my head on how to dig into these data a little bit more completely and think about what they can tell us about how environmental variables constrain changes in biodiversity, and that ultimately might influence these functioning and patterns in ecosystems. And so while there's complex relationships amongst the differences in biodiversity change, what we see is that the gain and loss of species at small changes in streamflow are seeming to be really important, and that this is partially compensated by the relative abundance and the plasticity that occurs in the species that are shared amongst the different ecosystems. And so to kind of wrap this up, what we see in this system is that despite this really large temperature gradient, kind of stream flow is the dominant control on organic matter mass, its composition, and ultimately its stoichiometry. And I think there is highlights this important kind of interplay amongst the environment and its effect on biodiversity, and how that kind of mediates processes and functions that we care about, such as like the total attainable biomass or the nutrient sequestration of ecosystems. In this case, all the stoichiometries of the ecosystem organic matter pools were constrained relative to species and communities. And that we really highlight the role of multiple dimensions of biodiversity change and its influence on organic matter stoichiometry. And I think this is an interesting question. There seems to be not a lot of, maybe I'm missing it, but there seems to be not a lot of data kind of trying to look at data or studies trying to look at the relative influence of certain processes, such as intraspecific variability versus intraspecific variability on kind of this aggregate level. I think, Paul, you have a 2012 paper looking at macrophyte communities in lake in relation to land use. There was a paper, Sven Terlenu, looking at kind of the effects of light and phosphorus on changes in stoichiometry, and that was using the price equation. And I think this might represent an opportunity to kind of break in and make these connections within kind of a stoichiometric framework. Make these connections amongst changes in the environment and changes in things like nutrient availability or temperature, and how that might mediate biodiversity and function. And when we think about biodiversity, in this case, in multi-dimensions, and how those dimensions interact to potentially create some resistance or resilience of certain ecosystem functions in the face of global changes. And so with that, I certainly thank you for your attention. I'd love to take any questions, and certainly thank the co-authors, confidants, and support people and staff and the funding sources. Thanks. I'm glad to see I wasn't talking at a blank screen or anything. It's interesting giving such a presentation in such a quiet environment.
Yeah, so Jim, many thanks for giving that talk. It's really interesting. I've seen different versions of this Eisen study, and this was an interesting, I guess, aspect of the project I didn't know much about. If there's questions, please jump in and either just ask them or feel free to write your questions in the chat. And Jim, you'll be around for a few minutes to.
Yes.
Yeah. The first thing I would just say is when I at the end of your talk where you're talking about biodiversity connecting to the sort of community level stoichiometry did remind me of my study, but I would just say honestly, I have no idea what I was doing when I was working on that. I had similar kinds of questions, but I had no idea. So I think what you're doing is really far more sophisticated, and I just would commend you on that. I'd like to see more of that. It'd be cool to see how that works with the data that we had.
Yeah, I thought when I first saw your study, I thought, oh, this might be an interesting, because I know I'm trying to remember if you were exact results, if you found like that interspecific or within stoichiometric, within species variability was less important than amongst species variability. I'm trying to, might have it backwards.
So we basically just sampled macrophytes in a defined area and then just biomass averaged the elemental composition and said this is the community CNP ratios. And then is that being driven because of within species or between species differences? And the thing that the journal didn't let us publish was we found like a really strong correlation between the biomass dominant plant. So like some plants are like 90% of the biomass. So whatever the community stoichiometry was basically that plant. And so if like you've got rid of that one plant, you would dramatically change. But you could change, like 3 or 4 of the other species, whether they were there or not, or their biomass. But I don't think it would actually change the overall. But we weren't able to sort of partition out like presence absence of the species versus the relative abundance and that kind of stuff. And it seemed to me like there was a lot more to it and a lot more complexity that we just didn't really quite know how to handle or deal with in that shorter paper.
Yeah, cool. Yeah, it would be interesting to see if the price equation could, potentially elucidate and lighten some of that.
There's a question in the chat. I don't know, Jim, if you just want to read that out.
Yes. So do I understand correctly that species and communities have quite a lot of stoichiometric variability, but if you add up the stoichiometries of the biomass in the entire system, there's almost no stoichiometric variability? And is this within one ecosystem over time or between similar types of ecosystems? Yes. So, and this was Camille asking the question. And so, yes. So when you look within a species or within a community, we see kind of a large measure. There's relatively large relative variability within at those levels. But when you add up all the biomass stoichiometries, that they exist within a much narrower range of variability. So not a lot of stoichiometric variability, yes. This is within, 11 different streams. Probably forgot to mention that are kind of in this similar watershed that range from 5 to 30 Celsius in temperature.
So just following up on that and thinking about Paul's stuff, because I find that pattern fascinating, and sometimes it depends on what trait you're looking at, right? So some traits can be, way more variable at those levels than others. But I was thinking in yours, and I think you talked about with the price equation, sort of the changes in diversity, right? The changes in diversity kind of have a neutral effect, and I was wondering how that relates to, like, a lot of your communities seem to be dominated by sort of one organism, and kind of what Paul was saying. Yeah, if you vary a lot of those down here, you're not going to see that on the ecosystem level. Is that?
Yeah, I think there's certainly that is certainly at play a bit. In some of the, especially the systems with higher standing organic matter pools, they're often dominated by these bryophytes that hold, that are dominating the community. And once you lose those, that's, I think, where lots of those effects are manifesting in terms of the decrease in stoichiometry and that.
So I have,
yeah.
Sorry. No, go ahead. No, go ahead.
I'm trying to remember where I was going. Maybe I'll come back to it, but what did you have?
Okay, no, I'm so sorry I cut you off. Yeah, no, I find my whole PhD was about variation, so I think about this all the time. It's really cool. And I know that you all measured a lot of other ecosystem functions. So do you have any feel for whether this pattern holds that way? Like, are some of the other functions way more variable potentially than just organic matter pools or stoichiometry?
Yeah, I'm trying to think if there's. I think. Yeah, I mean, for some of the other functions, this informed a lot of the future chapters that I did, and actually in some regards, like once we found this that temperature really was kind of overridden by flow, I subset the streams that I was intending to, for instance, measure secondary production on, because I didn't want to try to confound these effects of total organic matter mass and temperature that were, that flow was really the dominant driver, rather than temperature itself. So we actually subset the streams to focus on similar primary producer communities, and get around that kind of these compositional differences that exist.
Yeah, I really love all the work that came out of you guys' stuff, just because it's not just temperature, right? Like global change is not just temperature. That's but anyway, thanks.
Yeah.
I mean, this is really really fun to see these results.
And following up on Camille's chat, so the 3 dots in that figure where we're looking at the variability in stoichiometry represent carbon to nitrogen, carbon to phosphorus, and nitrogen to phosphorus stoichiometry for each of them. That clarifies. And Clay, I remembered what I was going to say regarding the kind of the dominance and loss of really important species. So the interesting and kind of, I think, cool thing about the Price equation is that the total effects of each of those components is the summed effect of a species contribution towards that component. And so you could break down each of those components even further and look at what are the relative effects of a single species on each of those kind of higher level aggregate partitions, in theory.
Yeah, that would be awesome. I might need a math tutorial from you for that, but yeah, it'd be fun to apply it to other stuff like Paul's data sets and stuff and see what happens. So yeah.
Totally.
I'll jump in with a question.
All right, Karen.
Hi. I think at one point you mentioned that it was different to distinguish between the effects of nitrogen availability and flow on stoichiometry. Is that correct? I was wondering if that was because you didn't have a large gradient in nitrogen, or
yeah, I think the gradient that does exist is really small in these systems. They're really like the N to P ratios that we're seeing are sometimes below 1. So they're actually really nitrogen limited and relatively, I think, actually, let's think. I have, there it is. So this is the kind of, the DIN to D SRP ratios that we observed were between like 5 and 0, less than 1, and DIN range is relatively low because of that. Sometimes, occasionally below detection, which is odd. And so, yeah, this was something that I think that's why you don't see DIN driving the seed-end ratios of the ecosystem biomass, and you kind of have that compositional effect that's much more important in terms of species traits and species composition.
That makes sense. Thanks. I was also wondering, this is kind of a basic question, but I haven't thought that much about flow. By what mechanisms do you think that's influencing stoichiometry?
Yeah, so I think what is happening is that the patterns of stream flow over kind of on longer timescales kind of sets the stage for the existence or not of these larger kind of complex plant species, macrophytes, bryophytes, and they have a lot more structural tissue. And so those things get knocked down by these floods at high levels of stream flow. And that's really the dominant kind of that shift between forest and grassland is kind of the terrestrial analogy is, if you have high levels and highly frequent disturbances, you probably have high levels of variability in flow, and that keeps the communities more similar to grass.
That makes sense. Thanks for the great talk.
Thanks.
All right, any more questions? You may have all just heard my dog barking, so sorry about that. She has a lot of bark, but not much bite, apparently. And anything else? So yeah, no, but thanks again, Jim. And I look forward to seeing more of your work coming out. And let's talk about some of this at some point if
Yes.
want to follow up on it. It's really interesting. It's good.
Thanks. Yeah. Thanks for the invite, and thanks for putting on the series. It's been excellent. Proud to be a part of it.
All right. Thanks. All right. Thanks everybody for coming.
Thanks. It was really good.